Hello everyone and welcome back to the channel. In this episode we're going to discuss about the microbial, catabolic, and anabolic processes. Catabolic process is a metabolic pathway that releases energy by breaking down complex molecules into simpler substances. The opposite of catabolic process is an anabolic process which uses energy to synthesize complex molecules from simpler ones. Glycolysis and Krebs cycle are two important catabolic processes that are used by microorganisms to generate energy. Both processes occur in the cytoplasm of cells and both break down glucose, but they do so in different ways and at different stages. In glycolysis, glucose is broken down into two molecules of pyruvate. This process does not require oxygen and it generates a small amount of ATP and NADH, which can then be used for other metabolic processes. The Krebs cycle, also known as the citric acid cycle or the tricarboxylic acid cycle, takes place after glycolysis. It is a series of chemical reactions that completely oxidizes the acetyl group of pyruvate to carbon dioxide and water. This process requires oxygen and generates ATP, NADH, and FADH2, which can be used in the electron transport chain to generate a proton motive force that drives ATP synthesis. The main difference between glycolysis and the Krebs cycle is that glycolysis occurs in the cytoplasm and does not require oxygen, whereas the Krebs cycle occurs in the mitochondria and requires oxygen. Additionally, glycolysis only partially oxidizes glucose, whereas the Krebs cycle completely oxidizes the acetyl group of pyruvate to carbon dioxide and water. Although the Krebs cycle is a central metabolic pathway, it is not universal among all living organisms. In fact, many bacteria and archaea cannot carry out the full cycle due to the absence of some key enzymes. However, these organisms have evolved alternative pathways to generate energy from glucose and other carbon sources. For example, some bacteria such as Escherichia coli can carry out the Entner-Doudoroff pathway, which is an alternative to glycolysis. Others can utilize the pentose phosphate pathway to generate NADPH, which is used in biosynthetic reactions. And still others can ferment sugars without producing alcohol or carbon dioxide through a process known as the Stickland fermentation. To learn more about the catabolic and anabolic processes, let's start with glycolysis, which is the breakdown of glucose. As mentioned earlier, glycolysis occurs in the cytoplasm of cells and is the first step in the catabolism of glucose. During glycolysis, one molecule of glucose is converted into two molecules of pyruvate. This process generates a small amount of ATP and NADH, which is a reducing equivalent that can be used for other metabolic processes. There are 10 steps involved in glycolysis, and each step is catalyzed by a specific enzyme. The first step is the phosphorylation of glucose to glucose 6-phosphate by the enzyme hexokinase. This reaction requires one molecule of ATP. Glucose 6-phosphate is then isomerized to fructose 6-phosphate by the enzyme phosphoglucose isomerase. The third step is the phosphorylation of fructose 6-phosphate to fructose 1,6-bisphosphate by the enzyme phosphofructokinase. This reaction also requires one molecule of ATP. Fructose 1,6-bisphosphate is then cleaved into two 3-carbon sugars, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate by the enzyme aldolase. The next step is the isomerization of dihydroxyacetone phosphate to glyceraldehyde 3-phosphate by the enzyme triose phosphate isomerase. All the glyceraldehyde 3-phosphate molecules are then oxidized by the enzyme glyceraldehyde 3-phosphate dehydrogenase. This reaction generates one molecule of NADH per molecule of glyceraldehyde 3-phosphate and one molecule of ATP per molecule of glyceraldehyde 3-phosphate. The sixth step is the phosphorylation of 1,3-bisphosphoglycerate to 3-phosphoglycerate by the enzyme phosphoglycerate kinase. This reaction generates one molecule of ATP per molecule of 1,3-bisphosphoglycerate. The seventh step is the transfer of a phosphate group from 3-phosphoglycerate to 2-phosphoglycerate by the enzyme phosphoglycerate mutase. The eighth step is the removal of water from 2-phosphoglycerate to form phosphenol pyruvate by the enzyme enalase. The ninth step is the transfer of a phosphate group from phosphenol pyruvate to ADP to form ATP by the enzyme pyruvate kinase. This reaction generates one molecule of ATP per molecule of phosphenol pyruvate. Finally, pyruvate is formed from phosphenol pyruvate by the removal of one carbon dioxide molecule. As mentioned earlier, the net yield of glycolysis is 2 molecules of ATP, 2 molecules of NADH, and 2 molecules of pyruvate per molecule of glucose. After glycolysis, if oxygen is present, pyruvate is transported into the mitochondria where it is oxidized in the Krebs cycle. Pyruvate is first converted to acetyl coenzyme A and carbon dioxide by the pyruvate dehydrogenase complex. This reaction generates 1 molecule of NADH per molecule of pyruvate. Acetyl coenzyme A then enters the Krebs cycle where it is completely oxidized to carbon dioxide and water. The Krebs cycle consists of a series of 8 reactions that occur in the mitochondrial matrix. In the first reaction, acetyl coenzyme A condenses with oxaloacetate to form citrate by the enzyme citrate synthase. Citrate is then isomerized to isocitrate by the enzyme aconitase. Isocitrate is then oxidized by the enzyme isocitrate dehydrogenase, generating 1 molecule of NADH and 1 molecule of carbon dioxide. The fourth step is the oxidative decarboxylation of alpha-ketoglutarate to succinyl-CoA by the enzyme alpha-ketoglutarate dehydrogenase. This reaction generates 1 molecule of NADH and 1 molecule of carbon dioxide. Succinyl-CoA is then converted to succinate by the enzyme succinyl-CoA synthetase. This reaction generates 1 molecule of GTP, which can be converted to ATP. Succinate is oxidized to fumarate by the enzyme succinate dehydrogenase, generating 1 molecule of FADH2. Fumarate is then hydrated to malate by the enzyme fumarase. Finally, malate is oxidized to oxaloacetate by the enzyme malate dehydrogenase, generating 1 molecule of NADH. As mentioned earlier, the Krebs cycle requires oxygen because the electron carriers NADH and FADH2 must be reoxidized to NAD+ and FAD for the cycle to continue. The NADH generated during the Krebs cycle can be used in the electron transport chain to generate a proton motive force that drives ATP synthesis. The overall equation for the complete oxidation of 1 molecule of glucose in the presence of oxygen is as follows. C6H12O6 plus 6O2, 6CO2 plus 6H2O. The number of ATP molecules generated from 1 molecule of glucose is actually difficult to determine because the energy released during the catabolism of glucose is not entirely converted into ATP. However, the minimum theoretical yield of ATP from the complete oxidation of glucose is 38 molecules. This includes the ATP generated during glycolysis, the Krebs cycle, and the electron transport chain. In addition to ATP, the catabolism of glucose also generates reducing equivalents in the form of NADH and FADH2. These molecules can be used in anabolic processes to synthesize complex molecules from simpler precursors. Anabolic processes are those that build up complex molecules from simpler ones, and they are essential for growth and development. Some examples of anabolic processes include the synthesis of amino acids, fatty acids, and nucleotides. In order for anabolic processes to occur, energy must be supplied. This energy can be obtained from the catabolic processes that break down complex molecules, such as glucose. The energy released during catabolism is stored in the form of high-energy bonds in molecules like ATP. Anabolic processes use the energy stored in these molecules to drive the synthesis of complex molecules. For example, the synthesis of amino acids from simpler precursors such as ammonia and alpha-keto acids requires energy that is supplied by ATP. Similarly, the synthesis of fatty acids from acetyl-CoA and other precursors requires energy that is supplied by reducing equivalents such as NADPH. The link between catabolism and anabolism is important for maintaining cellular homeostasis. When there is an excess of nutrients, catabolic processes break down these nutrients, and the energy is used to generate reducing equivalents and ATP. These molecules are then used to drive anabolic processes, which synthesize complex molecules that are needed for growth and development. When nutrient availability is low, catabolic processes are slowed down, and the cell relies on its stores of ATP and other energy molecules to drive anabolic processes. Thank you for watching, and I hope you found this video useful. If you have any questions or comments, please leave them below. And if you like this video, please give it a thumbs up and subscribe for more microbiology videos. See you next time.